Hi everyone. Today I am going to discuss about nitrogen cycle and symbiotic nitrogen fixation. Nitrogen. Nitrogen is the constituent of amino acid, nucleic acid and amino sugars. In atmosphere, nitrogen gas is about 78%. Valency state -3 to +5. Nitrogen is required by the plant in large quantity, but it is not available to them in any usable form. This is due to the high stability of the nitrogen triple bond, that is N triple bond N. Plants and animals require nitrogen, but the ability to fix nitrogen is restricted to some microorganism, RK, and symbiotic associations. All habitats require fixed form of nitrogen. The photosynthetic activity of the plants could not take place without the availability of the fixed nitrogen provided by the microorganism or by the synthetic fertilizer. Reservoirs of the nitrogen are dead and living organic matter, humus, soil organic matter, and inorganic ions like ammonium, nitrite, and nitrate. Nitrogen cycle. The conversion of atmospheric nitrogen into nitrogenous compound by microorganism is called biological nitrogen fixation. There are three main stages in the nitrogen cycle, that are ammonification, nitrification, and nitrate reduction and denitrification. Okay, so these are the stages in the nitrogen cycle. First, we can move to the ammonification. The conversion of organic nitrogen to the ammonia is called ammonification. The amino acid formed by the protein degradation are acted upon by the microorganism and it leads to the formation of ammonia. It is called ammonification. Many plants, animals, and microorganisms are capable of ammonification. Some of the examples for the ammonifying organism are Clostridium, Micrococcus, Proteus, etc. There are two stages in the ammonification. First one, proteolysis. Second one, amino acid degradation. Proteolysis is the breakdown of protein into simpler forms. Example for the proteolytic bacteria are Pseudomonas, Proteus, Bacillus, and Clostridium. Soil actinomycete and many fungi are also proteolytic. They secrete the enzyme protease which convert the protein into peptides which are then attacked by the enzyme peptidase to form amino acid. That is, the proteins are first converted to peptides and these peptides are acted upon by the enzyme peptidase to form amino acid. Second step in the ammonification is the amino acid degradation. The amino acid formed by the proteolysis undergo degradation to form ammonia. Example, alanine plus half O2 gives pyruvic acid plus ammonia. Here the enzyme alanine deaminase catalyzes the reaction. Second step in the nitrogen cycle is the nitrification. In nitrification, ammonia or ammonium ions are oxidized to nitrate ions and then to nitrate ions. The conversion of ammonia to nitrate is known as nitrification. The conversion of ammonia to nitrite is carried out by nitroso-vibrio, nitroso-lobus, nitrospira, nitrococcus and nitrosomonas. And the conversion of nitrite to nitrate is by nitrobacter, nitrococcus, nitrospira and nitrospina. Okay, so these are the microorganisms involved in the nitrification. And the final step is the nitrate reduction and denitrification. Denitrification means the conversion of nitrates to nitrites and finally into nitrogen gas. Several heterotrophic bacteria are capable of converting nitrates to ammonia. Example, Thiobacillus denitrificans, Micrococcus denitrificans, Hyphomicrobium, Vibrio, Pseudomonas, etc. The denitrification process is completed by various steps. That is, 2HNO3 gives 2HNO2, then 2NO, then N2O, and finally N2. Next, we can move to the fixation of molecular nitrogen. Nitrogen is the important nutrient of the plants. The atmospheric nitrogen made available to the plants by a process called nitrogen fixation. Fixation of nitrogen is by three processes. First one, physical nitrogen fixation. Second one, chemical nitrogen fixation. And third one, biological nitrogen fixation. Physical nitrogen fixation is also called atmospheric nitrogen fixation. Then the chemical nitrogen fixation means the conversion of molecular nitrogen to the nitrogenous form through man-made chemical methods. Example, Haber-Bosch process. Here, high pressure and temperature are provided to produce ammonia from nitrogen. That is, N2 plus 3H2 gives 2NH3 by giving high pressure and temperature. Here, 400 degrees Celsius temperature is used and 200 atm pressure. Okay. Then third type of the nitrogen fixation is the biological nitrogen fixation. The atmospheric nitrogen converted to nitrogenous compound by microorganism is called biological nitrogen fixation. Prokaryotic microbes reduces the atmospheric nitrogen to the ammonia by three ways. First one, symbiotic nitrogen fixation. Second one, non-symbiotic nitrogen fixation. And third one, associative symbiotic nitrogen fixation. Non-symbiotic nitrogen fixation occurs in free living organisms in the soil. Large number of the bacteria and cyanobacteria are included in non-symbiotic nitrogen fixation. And the associative nitrogen fixation found in association with the roots of grasses and cereal plants. Here, nodules are formed. Example for the associative nitrogen fixers are Azospirilla. Okay. Some of the examples for the nitrogen fixing bacteria are Azospirillum, Azotobacter, Bacillus, Beigernica, Chromatium, Clostridium, Chlorobium, Desulfovibrio, Vibrio, Rhizobium, Rhodospirillum, Rhodomicrobium, Pseudomonas, and Thiobacillus. Symbiotic nitrogen fixation. It is the important type of nitrogen fixation, and it is carried out by the microorganism which causes a tumor-like growth in plant. And this tumor-like growth in plant is called nodule. Symbiotic nitrogen fixation is accomplished by the bacteria of the genus Rhizobium in association with legumes. Azorhizobium and Bradyrhizobium are also able to carry out symbiotic nitrogen fixation. These are able to convert atmospheric nitrogen to the ammonia by nitrogen fixing mechanism. Rhizobium is a gram-negative rod-shaped bacteria, and it is a fast-growing, free-living heterotroph. The process nitrogen fixation is dependent upon the enzyme nitrogenase. There are 3 stages in the nodule formation. First stage, recognition and attachment. Second stage, penetration and travel. Third one, bacteroid formation and development of mature nodule. First stage in the nitrogen fixation is the recognition and attachment. Plant root secretes a variety of metabolites. Plant root means legume plant root. So the rhizobia migrate towards and grow in the rhizosphere region. The flavonoid signals present in the plant leads to the exchange of recognition signals. Thus, Rhizobium species attaches to the legume plant root hair. Lectins, carbohydrate containing protein, also contribute in Rhizobium legume attachment. Rhizobium possesses a specific adhesion protein called adhesion. This protein is present on their surface. Adhesion is a binding protein that can bind with the calcium complexes present on the surface of the root hair. The genes involved in the nodule formation is called nodulin genes. Second stage in the nodule formation is the penetration and travel. Polygalactourinase released by the bacteria depolymerizes the plant cell wall, and it allows the bacteria to invert the plant root tissue. After the penetration, the root hair curls as a result of the node factor secreted by the rhizobium. So the curling is also affected by the indole acetic acid. Indole acetic acid is a plant growth hormone. So the curling is the result of the node factor and indole acetic acid. After the curling, the bacteria enter the root hair and induces the plant to develop a cellulose tube called infection thread. This infection thread extends inward to the root hair. The rhizobium cells spread within the infection thread, later move on to the underlying cells, and finally it releases to the cytoplasm. The node factor increases the cell division that leads to the root nodule formation. Bacteroid formation and development of the mature nodule. It is the final step in the nodule formation. When the bacteria released from the infection thread to the host cell cytoplasm, they get transformed into swollen irregular shaped branched structure called bacteroids. Bacteroid is surrounded by a plant derived membrane called peribacteroid membrane and form a structure called symbiosome. Symbiosome is the site of nitrogen fixation. Next, we can discuss about a protein called leg hemoglobin. Symbiosomes secrete a hormone which enables the production of leg hemoglobin. It protects the nitrogen fixing enzyme from oxygen. It is a unique protein similar to the hemoglobin found in the animal system. Because of the presence of leg hemoglobin, nodule appear pink in color. The heme component of the protein is synthesized by the rhizobium and globin comes from legume. Nitrogenase. Nitrogenase is an enzyme complex that is responsible for the nitrogen fixation. Nitrogenase producing bacteria convert atmospheric nitrogen to the ammonia. Nitrogenase enzyme is very sensitive to the oxygen. That is, nitrogenase enzyme has two coproteins. First one, dinitrogenase or FEMO protein, and second one, dinitrogenase reductase, that is FE protein. Dinitrogenase has a molecular weight of 2,20,000 and dinitrogenase reductase has molecular weight 64,000. Dinitrogenase is joined with one or two molecules of dinitrogenase reductase. The nitrogenase enzyme is an equilibrium mixture of FEMO protein and FE protein. The electron initially transferred to the dinitrogenase reductase, then dinitrogenase, then passes to the iron molybdenum cofactor, and finally to the nitrogen. N2 plus 6 electron gives 2NH3. Here, H2 is involved in this reaction. And also, reduced ferredoxin, coenzymes, cytochromes, and ATP are also required for the fixation. So, that's all about nitrogen fixation and symbiotic nitrogen fixation. Okay, I hope you got an idea about nitrogen cycle and fixation. Thank you.